we have one final group to look at, and it might be the one that you're most interested in because it's the animals. And of course, we're a bit biased being animals ourselves. When most people think of animals, they think of something cute and fuzzy with a spine. But that's a rather small percentage of the animals. We have a lot of invertebrates, things like insects and worms and so on. They make up the majority of this kingdom. So what are we dealing with here? Well, this is the last group of the eukarya. Remember, we've talked about the protista, the fungi, the plantae, and now we have our animals. As members of the domain Eukarya, they're made up of eukaryotic cells. As you know, eukaryotic cells have a nucleus and they have organelles. All animals are multicellular and they have specialized tissues for the most part. Now, the one exception would be the sponges. They're kind of on the cusp of what it means to be an animal, but most animals do have these specialized cells, specialized tissues that carry out specialized functions within the multicellular organism. Animals don't have a cell wall. We don't have a rigid cell wall made of cellulose like we see in plants or made of chitin like we see in fungi. Instead, we have proteins wrapped around the outside of the plasma membrane, mostly collagen. So collagen consists of proteins arranged kind of like ropes or cables, and they help support the cell. But the cell is far more flexible than we would see in a plant or in a fungus. Some animals, not all, but some, have a skeleton of some sort. They use minerals, usually mixed with collagen or other proteins, to create something a bit more rigid on the outside of the animal, perhaps. That's an exoskeleton. Think of an insect or a lobster. They have a hard shell on the outside. Or in the case of vertebrates like ourselves, we have an internal skeleton, an endoskeleton. I'm going to divide our discussion of the Animalia into 3 topics. Today we'll do a very brief introduction into what defines animals, what makes an animal. And we'll talk about some of the characteristics that we use to define the major groups. In the next topic, we'll talk about non-deuterostomes. This is a large group that includes most of the invertebrates. And then we'll talk about deuterostomes. This is vertebrates like ourselves and their close relatives. So what defines an animal? Well, let's start with sex. It's pretty important here. If you recall, in fungi, they spend most of their life cycle as multicellular haploid individuals. Plants alternate between being multicellular haploid and multicellular diploid. In animals, we spend most of our time in a multicellular diploid state. The haploid state is very short-lived, and it consists just of the gametes. So the life cycle is what you learned in health class. It's pretty straightforward. It's actually, I would say, the simplest of the life cycles, where we have multicellular diploid adults that create haploid gametes through meiosis. That would be the sperm and the egg, and the sperm and the egg come together in a process known as fertilization, or syngamy if you want to be really fancy. This creates a diploid zygote, and then that undergoes mitosis to create the next generation. Another thing that sets animals apart from other kingdoms is the fact that the vast majority of animals are ingestive. What that means is they take in food in big chunks. We're great examples of this. We take in food in big bites, we break it down in our digestive system, and then it's passed into the blood and it's transferred to all the cells throughout our body. Remember that plants and algae use photosynthesis to make their own food, and fungi secrete enzymes into their environment, break things down outside of their cells, and then soak that stuff up. Now, there are some animals that are absorptive. The majority are parasites. Think of things like tapeworms, but they're not generally secreting enzymes. Instead, they're living in an environment where stuff has already been broken down for them. Okay, so those are features that we might use to separate animals, distinguish animals from other groups. Now, how do we distinguish the major groups of animals from each other? The first thing we might look at is symmetry. Symmetry refers to the idea that you can draw a line, draw a plane through an object, through an organism, and divide it into two equal halves. Think of something that's asymmetrical. Imagine you find a rock. You find this lumpy, weirdly shaped rock. You can't split it into two equal halves. It has no symmetry. But now let's say you have something like a tennis ball. There are many, many ways, in fact, every single plane that passes through the middle of that tennis ball will divide it into two equal halves. It has symmetry. All right, we have a few examples here. At the top there for A, we have something called a sea anemone. We'll talk about them later. And you can see that it's got a long axis. What I mean by that is part of the body is longer. So it has one axis that's long, one axis, the width, that's shorter. We can cut a plane along that long axis, and so long as it passes right through the middle, we can cut that anemone into two equal halves. Same thing with the flower pot next to it. So long as we're cutting along that long axis, and we are running our plane of section through the middle, we can divide it into two equal halves. This is known as radial symmetry. So if we look down on these objects from above, we're seeing roughly a circle. And if we cut along the radius or the diameter of that circle, we can cut it into two equal halves. Now down the bottom, we have a lobster and a shovel for some reason, and we can cut each of these into two equal halves, but only along one plane, right through the middle. So right through the face and down the tail of the lobster, right through the middle of the shovel. There's only one plane of section that can cut these critters, or shovels, into two equal parts. That's known as bilateral symmetry. We see bilateral symmetry in animals that have a well-defined head. That sea anemone does not have a well-defined head. A jellyfish does not have a well-defined head. So things without a well-defined head are quite often radially symmetrical, and things with a well-defined head are bilaterally symmetrical. If something has a well-defined head, we say it is cephalized. We'll come back to that. So if we look at humans, humans are quite obviously bilaterally symmetrical. We can cut a human into two equal parts, but there's only one way we can do that. Right down the middle, right down the nose and through the middle of the body. Now I should note, I'm just talking about the outside of the human. Some of the organs are asymmetrical, they're off-center, but we can definitely take a human, cut it into two equal parts, but there's only one way we can do that. So here we've got a human that's divided in a few different planes. Again, that one plane, and it's called the mid-sagittal plane. We won't worry too much about that. But that one plane will cut the human into two equal parts. Bi means two. The other planes will not. Before we go any further, we'll take a very quick look at the phylogenetic tree for animals. We'll come back to this in more detail later. It is believed that the kingdom Animalia is monophyletic, and that's nice. That means that all of the known animals can be traced back to one common ancestor, and that common ancestor was the choanoflagellates. Now these are things that are still around. It's a type of protist. Remember that in evolution, ancestors don't have to disappear. They might become extinct, but it's a branching process. Choanoflagellates are these small, filter-feeding little protists, and they're found predominantly in marine environments, ocean environments. Now, these ancestral choanoflagellates were colonial. What that means is these cells were living together quite happily, but there was no specialization, so we don't have any specialized cells or specialized tissues. These choanoflagellates branched off into two broad groups. Now, another name for animal is metazoan. We have the true metazoans. EU means true. They have true, well-defined tissues that have different functions. A tissue is a collection of cells. And then we also have the parazoa, and they do not have true tissues. The parazoa are the sponges. Para means next to. Think of parallel. So they're next to the eumetazoans. They're not really quite animals, but they're very closely related. The eumetazoans split into two broad categories. We have the radiata. These are radially symmetrical. They're quite simple animals, as we'll see. And then we have the bilateria, which are bilaterally symmetrical. The bilateria split into what are called acoelomates. They don't have a body cavity. This is the cavity that surrounds the internal organs. They don't have it. And then we have another group that does have body cavities. And then within that group, we have what are called the pseudocoelomates. They have a body cavity, but it's not surrounded by something called mesoderm. We'll come back to that. Don't worry about that yet. We have the true coelomates. And then the true coelomates can be divided further into what are called the protostoma and the deuterostomes. Those are the groups we're going to focus on, and we are in that deuterostoma group. Again, I know this isn't going to make much sense just yet. I just want you to be aware of this tree. We're going to come back to it over and over and over again. Let's start at the beginning and take a look at the root of that phylogenetic tree. Let's take a look at the ancestor of all the animals. Again, it's called a choanoflagellate. It's a type of protist. It still exists. We're talking about single cells here that can live entirely on their own, depending on the species, or they can clump together. And when they're clumped together, we refer to that as a colony of cells. We don't consider this a multicellular organism because in multicellular organisms, the cells are highly specialized. If we take a look at our own bodies, we have muscle cells that are highly specialized. All they do is they shrink, they contract forcefully and generate force, tension, so that we can move around. We have highly specialized cells in the back of our eye, in the retina, that respond to photons of light so we can see. We have neurons, which are highly specialized. We have tissues that are made up of sets of specialized cells. And we have about 350 distinct tissues that make up our bodies. But that's not what we're seeing here. All the cells are the same. They are not specialized in any way. So on the right, you're seeing a colonial choanoflagellate. So we have, maybe 6 cells. I think there's one on the back there that we can't see. And they're connected together and they're attached to a stalk. Let's take a look at one of those cells. You can see each one has a flagellum. That's that whip-like structure. And then each cell also has something called the collar. And the collar is this region here. There's a bunch of little filaments that stick out from the body of the cell. So the body of the cell is below that. You can see the nucleus in here. That collar, those filaments are very sticky. They're covered in mucus. And what happens is that flagellum whips back and forth, and it creates a water current that draws water through that collar. Any food, bacteria for instance, that are in the water get trapped on that sticky collar and move towards the body of the cell, and then the cell engulfs it. And that's how it feeds. Now there is strength in numbers, so having other cells around you is a good thing, and these cells can share food. But again, the cells are all the same. There is no specialization of the cells. Now, how did something this simple become the first animal? Imagine we have a clump of these choanoflagellates. So we have this colony, and it's arranged in a solid ball. So you're seeing that down the bottom here in the first diagram. We have this ball of cells. The cells are all the same. You can see the flagella sticking out, and this is a ball that can roll and tumble around. Now imagine that develops into a hollow ball, as we see here. Incidentally, this is very similar to Volvox. If you remember, we talked about that briefly when we talked about algae. It has almost the same structure. We have a ball, a hollow ball of cells, and the cells have flagella. The flagella can move the whole ball around, and also the flagella can collect food that can be filtered out by the cells. Now we have, in the next case here, same thing, but we have a few cells now that are specialized just as reproductive cells. So this whole ball of cells is diploid. But those reproductive cells can undergo meiosis and produce gametes. And the gametes can swim off, find other gametes from another one of these colonies, and combine with those to produce zygotes and new individuals. Now let's say that we push in one side. So one side of this hollow ball, think of a tennis ball, is getting pushed in. Incidentally, that's something known as invagination. A vagina is a blind pouch, basically, and that's what you're creating here. You're creating a pouch that's a dead end. So the process of infolding or invagination is just where you take a ball like this and you push it in on one side. And that gives us something at the end here, the very last diagram down the bottom, that looks a lot like a gastrula. And we'll talk about that in a moment. But a gastrula is something we see in the embryonic development of all animals. In fact, all animals go through the stages that we see here that have been proposed for the very early development of the first animals. So now you can see that we've pushed in this side here, and now we have this cavity here, and this cavity can serve as a digestive cavity. So now you can imagine this organism swimming around using flagella to move food into that cavity in the middle. Those cells are going to secrete digestive enzymes, break down food, and then that's shared amongst all the cells. And what we have here is a simple animal. In fact, if we were just to add tentacles coming off down the bottom here, hanging off on the bottom, we would actually have a jellyfish. This is all there is to the jellyfish. A jellyfish has 2 layers of cells. It has this pouch on the bottom. It takes food into that pouch. There's enzymes that are secreted into that pouch, and that's how it feeds itself. So the stages that you're seeing here are stages that all animals go through during their very early development. You went through stages like this. For the jellyfish, they just don't move beyond that. The early stages of animal evolution are preserved in the early stages of embryo development in all animals. So let's have a look at that. You started out as a single cell, a zygote, and this was the product of your father's sperm and your mother's egg coming together. And that one zygote developed through mitosis into you when you consist of roughly 30 trillion or so cells. The zygote divided into 2 cells, they divided into 4 cells, they divided into 8 cells, etc. Those early divisions are known as cleavage divisions, and that results initially in a solid ball of cells. Now these divisions are pretty quick, and the cells don't have much time to expand. So we end up with lots and lots of small cells. As the divisions continue, this turns into a hollow ball of cells, and that's known as a blastula. So it's a hollow ball, like a tennis ball. There's a cavity in the middle, and it's referred to as a blastocele. Cele, C-O-E-L, means cavity. So this is a cavity within the blastula. Now what happens next is something called gastrulation. This is the process by which a gastrula is made. So what happens is one side of the ball gets pushed in. Again, that's referred to as infolding or invagination. Now once that happens, we have 2 layers of cells. So you're seeing that in the final diagram here. This outer layer, shown in blue, is known as the ectoderm. Ecto means outside. And the inner layer, the layer that's being pushed in, is known as the endoderm. Endo means inside. This is the gastrula. That hole where things got pushed in is known as the blastopore. And that cavity that's formed within the endoderm is referred to as the archenteron. Archo means old. Think of archaeology, the study of old human stuff. The archenteron is the ancient gut. Enteros means gut. So what's happened here is we started with a hollow ball of cells that got pushed in, and that created a cavity. That cavity is this very, very primitive digestive system, this primitive gut. And in most animals, that's going to develop further and give rise to the digestive system. Those 2 layers that have developed, the endoderm and the ectoderm, those are referred to as germ layers. It has nothing to do with what we usually think of as a germ, which is something that's infectious. Unfortunately, the word germ has several meanings in biology, but here, those very early layers that develop in the very early embryo, they're referred to as germ layers. So we have those 2 layers. We have the ectoderm. The ectoderm, shown in blue here, is going to give rise to the skin and also to the nervous system, as we'll see. Then we may or may not have a 3rd layer developing between the endoderm and the ectoderm. This is referred to as the mesoderm. Meso means middle. If this layer exists, it doesn't exist in some of the very simple animals, things like jellyfish. But if this layer exists, it gives rise to muscle and connective tissue. And connective tissue would include an endoskeleton, the kind of skeleton that we have. The endoderm, that innermost layer that lines that archenteron, that ancient gut, gives rise to the lining of the digestive system. Again, the goal of this topic is to introduce you to some important concepts that we'll return to as we talk about animals more. Another important concept that's used to distinguish different groups of animals is how those early divisions, those cleavage divisions, occur. Cleavage, once again, refers to the divisions that take us from a zygote to a solid ball of cells, and then to a hollow ball of cells known as the blastula. In the top diagram here, you're seeing something known as spiral cleavage. So we're starting with a 4-cell embryo, and it's dividing to give rise to 8 cells. And you can see that the cells are all offset. If we look at this from the top view, we can see that all the new cells are shifted over a bit to the right. That's spiral cleavage. Down the bottom, we have something called radial cleavage. And this is where the new cells sit directly on top of the old cells. Another concept we'll address briefly right now is the idea of body cavities. And I mentioned before that body cavities are the spaces within your body that contain internal organs. What you're seeing here are the body cavities in a human. So in yellow, we have the dorsal body cavity, and it contains the brain and the spinal cord. More importantly, though, we're going to focus on what's shown in red. That's the ventral body cavity. Dorsal means towards the back. Ventral means towards the front. Or if we're talking about a quadruped, something that walks on 4 limbs, it's towards the bottom. It's towards the tummy of the organism. Within the ventral body cavity, you have most of your internal organs, your visceral organs, your heart, your lungs, your stomach, your intestines, your bladder, etc. Now that ventral cavity is divided into subcavities. We have the abdominal pelvic cavity, we have the pleural cavity that contains the lungs, we have the pericardial cavity that contains the heart, etc. But really our focus right now is just this idea that you have this ventral cavity, this large cavity towards the front of your body that contains the majority of your visceral, your internal organs. Not all animals have body cavities. Some of the simpler animals, things like flatworms that we'll talk about in our next topic, are acoelomates. That means without a cavity. So again, a coelom is a cavity, a space. If you see that at the beginning of a word, generally means without. So they do not have a body cavity. If we look at these flatworms, they are solid pretty much all the way through. So they have a layer of skin, essentially, that's derived from that ectoderm from the very early embryo. And directly underneath that, they have mesoderm, which contains some simple muscle cells and some connective tissue. And then inward from that, they have their digestive tract, which came from the endoderm. But they don't have a cavity. Then I'm going to skip down to the bottom for a second. We have something called a coelomate, which has a proper coelom, a proper body cavity. We've got an earthworm here, but we would fit into this group as well. So you can see that we have this outer layer, that would be our skin, and it's derived from the ectoderm, that early embryonic layer. And then beneath that, in red, we have the mesoderm. That would be muscle and connective tissue, our skeleton as well. And then within that, we have the digestive tract. So your intestines, your stomach. The innermost layer of your intestines and stomach is derived from endoderm. The outer layers of your intestine and stomach are derived from mesoderm. And between your intestines and your stomach is a cavity. There's a space. And then there's more mesoderm-derived tissue that surrounds the inside of the body wall. So we have these spaces. There are spaces between your inner organs and the body wall. Your intestines and stomach are very slippery and they slide past each other. They wiggle around in this space. But that space is entirely lined by tissue that's derived from that middle layer, the mesoderm. Now in the middle, I haven't addressed these pseudocoelomates yet. Pseudo means false. They have a body cavity, but it's not considered a proper body cavity. And the reason for that is the cavity is not lined completely by tissue that's derived from mesoderm. So we have our digestive tract in the middle. It's completely derived from endoderm. And then there's a space. And then we have our mesoderm and then our ectoderm surrounding that. But the outside of the digestive tract doesn't contain any layers that are derived from mesoderm. Again, I realize some of this is maybe going to go over your head right now, but we will come back to this again. One thing we haven't talked about is where that body cavity comes from and how the mesoderm develops. There's two ways that it can develop. We have something called schizo or schizocoelous development. Let's break this down. What does that mean? You've probably heard of schizophrenia before. That is a splitting of the mind. Schizo or schizo means to split. Phrenia means the mind. Your mind splits, and that little voice in your head that you recognize as your conscience, you start to think of it as something different. You start to think you're receiving messages from the outside world, etc. That's beyond this course. This is not a course on psychology, of course. But schizo means to split the coelom, to split the body cavity. So what happens is we have mesoderm developing as a solid mass between the ectoderm and the endoderm, and then it splits and it forms a cavity. It forms a coelom. And then what would happen next, it's not shown here, but this would progress in both directions. So that mesoderm with a space in the middle would develop like this, and then we'd end up with a body cavity that was completely lined with mesoderm. Now the other way that cavity lined by mesoderm can develop is a process known as enterocoelous development. Entero refers to the gut, the digestive system. And here what happens is we have these little out pockets, these little folds that come off of the archenteron, that ancient gut. And you can see at the top here we have these cells shown in red that are becoming mesoderm. And they're going to pinch off, these little pockets are going to pinch off and form cavities. And they're going to move around and fill in this space. Again, the reason this is important is because it helps us identify major changes in development that help us distinguish different groups of animals. These are differences in development that appeared very, very early on in the ancestors of these animal groups. Okay, let's put this all together. We have two broad categories, two big groups of coelomate animals. Again, coelomates are animals that have a proper body cavity that's lined by mesoderm, and that's the majority of animals, ourselves included. These two groups are known as the protostomes and the deuterostomes. So looking on the right here, one of the first characteristics we can use to distinguish between these two groups is cleavage and how it occurs. Protostomes have spiral cleavage. Deuterostomes have radial cleavage. Now it also mentions in that diagram that the cleavage for protostomes is determinate and the cleavage for deuterostomes is indeterminate. What does that mean? Well, have a look at that first figure for the protostomes and notice that the cells on top are quite a bit smaller than the cells on the bottom. At this point, this really early point, where we just have 8 cells in the embryo, those cells are already starting to specialize somewhat. They're already having their fate determined, basically. So the cells are already a bit different at this point. If we look at the deuterostome figure at the top there, where we have indeterminate development, those cells all look the same. They haven't accepted a specific fate yet, and we'll come back to that later. Okay, the next thing that we can use to distinguish between protostomes and deuterostomes is how the coelom is formed. So in protostomes, we have this schizocoelous development or schizocoelous development, where we have this massive mesoderm that forms between the ectoderm and endoderm, and then that splits to form a body cavity. And in the deuterostomes, we have this ontrocoelous development where the mesoderm kind of forms this pocket off of that archenteron, and then that gives rise to the body cavity. Finally, down the bottom here, something we haven't talked about is what happens to the blastopore. So remember, we have a hollow ball of cells, that's the blastula. One side gets pushed in, and that forms a hole, that forms an opening. In the protostomes, that blastopore gives rise to the mouth. The archenteron eventually will open up on the other end as well, and that gives rise to the anus. In deuterostomes, it's the other way around. The blastopore becomes the anus, and a second opening forms later, and that becomes the mouth. Proto means first, stome means mouth. Deutero means second, and again, stome means mouth. So protostome, the blastopore becomes the mouth. Deuterostome, the blastopore becomes the anus. The mouth develops secondarily. We are deuterostomes. We'll put all of this together by looking at the development of something known as Amphioxus. This is a small little creature. It's very closely related to an ancient ancestor of ours. It's a deuterostome. It looks like a little fish, but it is quite different. Again, we'll come back to that. But let's look at its development. So up in the top there, in the top left, we have our blastula. It's a hollow ball of cells. And you can see it gets pushed in on one side, and that gives rise to our endoderm. There's also some tissue there shown in green. That's going to become the notochord. And now that's a stiff rod that runs down the length of this animal. Again, we will come back to that. But what you can see happening here is we have the development of 2 layers initially, and then on the second row there at the right, so I'm looking over here, we can see the development of some mesoderm shown in red, and it's forming as a pocket off of the archenteron, and then that's going to form the coelom. That's going to form the body cavity, and you can see that here. So the body cavity is going to wrap around all the internal organs. The blue on the outside, that's the ectoderm. It's giving rise to the skin, but also if we go back a little bit here, we can see that it's also giving rise to this purple layer, which is going to fold inward and become the neural tube here, which is the spinal cord. And it is a tube, it's hollow in the middle, same case in us. But again, we're going to come back to all of this. As a quick preview, these are the groups of animals we're going to talk about. We'll talk about the porifera, which are the sponges, the cnidaria, which are the jellyfishes and anemones. We'll talk about the platyhelminthes, which are flatworms. We're going to skip a few groups because there's so many groups. We'll talk about the mollusca, the mollusks, snails, and also squids and octopi. We'll talk about the annelida, which are things like earthworms, the nematodes, which are other worms. We'll talk about the arthropods, which is the biggest group of animals. That would be things like insects and crustaceans. We'll talk about the echinoderms, starfish, sea urchins, and we'll talk about the chordates, which is the group that includes vertebrates. It includes us. Now, I don't know who this woman is here. I think I stole her off an Amazon ad, but she's got a very simple cnidarian on her head, and she is a chordate. But we'll cover that, and we'll cover everything in between. Our concepts here: animals, metazoans, are a monophyletic group of multicellular heterotrophic eukaryotes. Remember that heterotrophic means that they can't make their own food. So things that are photosynthetic are autotrophic. Heterotrophic organisms have to take in food from their environment. Animals don't have cell walls. Instead, they have a proteinaceous extracellular matrix, which is a really fancy way of saying that they have this layer of protein goo around the outside of the cell that helps protect them. But it's not rigid, it's more flexible. Those proteins also act as a glue to glue cells together into a multicellular organism. Choanoflagellates are thought to be the ancestor of animals. So these are protists, they still exist, as I've mentioned, and these cells are identical to the cells that make up sponges, as we'll see in our next topic. Colonies of choanoflagellate-like cells are thought to have formed into hollow balls, undergone invagination where one side of the ball is pushed in. That gave rise to a gastrula, and this would have been, a free-swimming, free-living animal that would have sucked food into that little pouch in the middle of the gastrula. That's how it would have lived, that's how it would have fed itself. But we still see these stages in all animals as they develop. Cleavage divisions of the zygote generate a hollow ball of cells, and that's referred to as the blastula. That undergoes gastrulation. That's where one end is pushed in to form this little pouch inside that ball. Gastrulation results in 2 or 3 primary germ layers. So the first process of gastrulation, where one end of that ball gets pushed in, is going to form the ectoderm and the endoderm. And we say that that's a diploblastic condition. Di means 2, so we have 2 layers. And then depending on the animal, we may develop a 3rd layer. This happens in most animals, all but the simplest animals. And that 3rd layer is the mesoderm. That is a triploblastic condition, tri meaning 3. In most animals, we have a coelom, a body cavity that develops. A true coelom, a true body cavity, which is found in a group of animals known as the coelomates, is going to be completely lined by tissue that's derived from that middle germ layer, from the mesoderm. And again, there's two ways that can appear: schizocilous or enterocilous. Schizocilous, we have a mass of mesoderm that just splits and forms a cavity. Enterocilous, we have these little pockets, these little pouches that come off of that central pouch, that archenteron, and form the body cavity. Animal classification is based on these concepts that we've just covered. It's based on symmetry. It's based on how many germ layers we have in the early embryo. It's based on the type of cleavage, the type of coelom, and how it develops, and the fate of the blastopore. Does it become the mouth, or does it become the anus? Finally, here's our terminology. These are terms we're going to be returning to over and over again in the next two topics. So once again, if this doesn't quite make sense yet, it's okay. We're going to get there. We're going to go over it again. I just wanted to introduce you to these broad concepts.